Hi everyone, my name is Valentin. I'm a biologist, not a computer scientist, and I'll be telling you how I use machine learning in my research to study the insect brain. And throughout the presentation, I'll be telling you mostly about this insect. It's called Megalopta genalis. It's a bee that lives in South America, in Panama. And what's interesting about it for us is that it's nocturnal and it's able to navigate in the dark in a very cluttered environment in the tropical forest. And so we want to understand how it achieves that. And in general, we want to understand how insects navigate. To answer the question, we want to look at their brains. And we want to look in particular at this small region at the center that we call the central complex. And it's a region that's composed of 4 neuropils. These are just synaptic dense regions that process different kinds of information. And why it's interesting is that we know that some parts process information related to the speed of the animal, others to the direction that the animal is traveling in, and another one that integrates both sources of information to produce a steering decision. So it tells the animal where to go and how to go there. And we want to understand how this works in this region of the brain on the circuit level. So to answer the question, as part of my PhD, I've been building upon this workflow that we try to automate as much as possible to process a lot of image data. And to acquire this image, we need to first dissect the central brain, and then we need to embed it. First, we need to stain it with heavy metals, and then to embed it in plastic resin. And so we obtain a tiny block of plastic that contains our stained tissues that now we can view under an electron microscope. And the specific kind that we're using is called a serial block face electron microscope, and it's simply a big microscope that's fitted with an ultramicrotome. And the idea is that we can then shoot an electron beam at the surface of the block, scan our image, and then we can have a diamond knife that comes and automatically cuts a thin slice off of the top. And if we repeat this process over and over again, we acquire images at different level of depth in our sample. And we can acquire an image stack that represents our region in 3D. And we have a multi-resolution approach, which is going to be important later, that was developed by Marcel, a previous PhD student in our lab. And so we scanned the entire region at low resolution. It's enough to see the backbones of the neurons. And then we have specific regions of interest that we scan at high resolution, where we can see all the smaller branches and even synapses. And then in the high resolution, we obtain this kind of image stacks. And here we can see different neurons flowing through the z-axis, and we can identify specific individual neurons that flow through it, and we can follow their path, and we can also identify synapses. And then if we do that for many neurons through our volume, we can obtain the full circuit in a region. And the big challenge is that we want to do that across many insect species. Now what I'm showing you is Megalopta, it's our proof of concept, but we have image data for more than a dozen insect species, and so the challenge here is that this is a lot of data to go through. And on average, the species have more or less the same size, and it's about 1.5 to 2 terabytes of image data that's processed, but the microscope actually produces more than this. And then some of the bigger insects, like the cockroach here, it's more than 3 terabytes of data. And so then the challenge is to process this data as efficiently as possible and taking as little time as possible. And the first step for this is to process the data, and in our case we need to align it because we work on such a fine scale, it's a 10 nanometers pixel size, that the smallest movements during scanning will cause the data to be really difficult to exploit, whether it's manually or automatically. So the first step, we use TrackEM2, it's a plugin that works on Fiji, and that uses a SIFT algorithm to detect feature of interest, which are really difficult to see here, but there's crosses there. And then we can detect the same features on an overlapping image. And then we can calculate the offset between these features, and from this offset, apply a transformation to our data. And here we use an elastic transformation. It's non-linear transformation. And the nice byproduct of that is that it will solve a bunch of deformation issues that we have from imaging our data. So then once we have that nicely aligned image stacks, we can start to extract biological information from it. and build upon our pipeline. And so as I was telling you earlier, we can nicely see the membranes of the neurons. We can see synapses here and there, and we can also see the mitochondria of the neurons in there. And that's the kind of information that we want to extract. And we do that in two ways. One, we have manual tracing, and here we're only tracing the skeletons of the cells, and it gives us the topology of the neurons, so where the branches go, how many branches, and so on. And that we still do manually. We use a software called CatMade, which makes it very easy to do this work, but it still takes a crazy amount of time. So now we want also to use automatic segmentation, and we want to do that in the high-resolution parts. I remind you here, that's the method of the multi-resolution approach. And so we do automatic segmentation where we can extract the smaller branches very, very fast. And we actually use the skeletons to both inform the segmentation and inform the proofreading of that segmentation. And so for the segmentation in the high-resolution part, we use UNet, and it was trained on all the data that you've seen so far that was extracted from megaloptites, our best data to date. And the input for the model is our raw image data and some manual segmentation that I annotated myself. And so for this, I took a few subsets of our data set and trained the model on it, and then evaluated it against skeletons on other parts of the data. And then we use a few steps of data augmentation. For this, we use a Python library called Gunpowder that just takes care of all of that. And the model is trained to output nearest neighbor affinity and local shape descriptors. So nearest neighbor affinity is really what we're interested in. It's the position of the membrane. We're detecting edges. And local shape descriptors, it's an auxiliary task that was developed by Jan Funke, a collaborator from Genelia, and his team. And it's a metric that is computed for every single voxel in the image that informs the model about the shape in 3D. And it was shown in that study that a model that can do both is better at detecting the membranes. And this architecture is also something that I just took from this paper. At the time, it was mostly because I didn't know better, but I'm glad that experts are telling me that it's the best way to go anyway. And so then, once I have my model that was trained and evaluated, I don't have this information here. I realize maybe you guys would like this kind of detail, but I have my big volumes and I can apply my model on it. So I obtain these predictions. That's our nearest neighbor affinity prediction. And then after a few steps of over-segmentation and agglomeration, I obtain some segmentation that's more or less satisfying. At least it's a very good starting point. But there are still some mistakes, and that's simply due to the model being imperfect, but also the image data being quite variable. So we have some false merges where two objects that should be separated are merged, and then some false splits where it's the contrary. And to solve this, we use Flywire. And Flywire is a platform that was created by people at Princeton University that lets users, through a user interface, visualize the data in 3D. So the segmentation in 3D and the segmentation also in 2D, and overlap it with the raw data, and they can come in and identify regions where there's a problem, solve it with a few inputs, and then there's a cluster in the background that calculates the best way to edit the segmentation graph in the background. So we have now this platform implemented thanks to the help of these people as well. And so we can have users come in and identify the regions where there's a problem, and then with a few inputs, solve it. And I realize this joke doesn't work here because you people probably understand graph theory. But so now, I've also been able to use this model on different species. On the left, you have a dung beetle, and on the right, you have a hawk moth, both coming from collaborators. And I could actually use my model that was trained on a completely different species and exclusively on that other species, and obtain some segmentation that was quite satisfying as well. This is still not perfect and it will need proofreading, but it's a very good starting point. And the interesting bit about that is that, well, for the dung beetle, we look at the same brain region and it's the same microscope, same imaging conditions and preparation. But the hawk moth, it's a different region. It's in the visual processing parts of the brain. And it's a different microscope, it's different imaging condition, different preparation, different voxel size, but the model still works very well. So it's able to generalize to data that was acquired in a different way. It's still electron microscopy, of course, but it's pretty good. And that means we will be able to use this model or a similar version of the model on others of our species. Oh, it's stalling like this. Yeah, so now we have this pipeline that's almost fully automated the process of extracting the data. And we can start to have some insights of how the brain works. And this is just the start. It's our proof of concept. I've used the model on the nodulus, which is that small part of the brain that encodes information related to speed of the animal. And I'm just going to show you some segmentation. So as I was telling you, we used first some skeletons. That was also work that was done before we had the segmentation. And these skeletons there allow us to identify cell types and thus to target our proofreading efforts. And it also helps the proofreader to just do it faster. And then with the segmentation, we've been able to identify different compartments in that region. One we know from other species and other studies encode rotational optic flow, so that's how fast the animal is turning. Others, translational optic flow, so how fast the animal is moving forward and backward. And then another one that we don't know what it does, but it's very interesting because we only found it in hymenopteran insects to date, so only in bees. And it hasn't been found in any other insects that are studied for their brain. And we believe and we hope that these cells encode route memory, so it would allow the animal to recall routes that it's taken in the past, either from the nest to resources or from the resources to the nest. And we know that is something that a lot of bees can do. And of course, the segmentation and the skeletons are not going to be enough to answer this question, so we need to add connectivity. And for this, we're working on implementing another model that does synapse prediction. That was again published by Jan Funke, our collaborator, and his team. And I'm working now with our new PhD student Griffin on implementing that. And so in the near future, we will have this full semi-automated pipeline that will let us exploit our massive amount of image data and hopefully answer this question of how insects navigates and how this brain region computes this information in the most efficient way possible. And with that, I want to thank you for your attention. Thank my supervisor, Stanley Heinze, all our collaborators, and the army of tracers and proofreaders that we have to help us with this work.